Dear learners, in this part of amylase inhibitor, we are going to study about different types of amylases, their sources and the mode of action. We shall also consider the digestion of starch in the human digestive system. At this stage, it will be appropriate at this stage to revise here the structure of starch. Starch is a polysaccharide made up of glucose. It is a storage polysaccharide present in plant seeds and tubers. Starch has a straight chain portion, it is called as amylose, which is made up of glucose units joined to each other by alpha-1,4 glucoside bond, and a branched chain amylopectin component with alpha-1,6 glucoside bond at the branching point. Amylose and amylopectin molecules are arranged in a highly organized pattern inside a starch granule. This organized structure of starch granule is destroyed during cooking by the uptake of water at high temperature, and it becomes more accessible to the action of enzymes. This has been shown in the figure showing the structure of the starch. Now, next coming to the amylases. Amylase represents a group of enzymes that catalyze the hydrolysis of starch into smaller carbohydrate molecules such as dextrins, maltose, which is a disaccharide, and ultimately to glucose. Two categories of amylases are denoted as endoamylases and exoamylases. The first type, endoamylases, are also called as alpha-amylase. The enzyme alpha-amylase, which is technically called as alpha-1,4 glucan 4-glucanohydrolase, and the EC number is 3.2.1.1. This is one of the major secretory product of salivary glands and pancreas, and they play a role in the digestion of starch as well as glycogen. Alpha-amylase is widespread among living organisms, especially in the digestive system of human beings and other animals as well. An alpha-amylase called ptyalin is produced by salivary glands, whereas the pancreatic amylase is secreted by the secretory cells in pancreas. It is also present in plants, especially in the germinating seeds, and is also secreted by many microorganisms. The alpha-amylase constitutes a family of endoamylases that catalyze the initial hydrolysis of large starch molecule into shorter oligosaccharides through the cleavage of internal alpha D14 glycoside bonds. Neither terminal glucose residues nor alpha-1,6 linkages are cleaved by alpha-amylase. The end product of alpha-amylase actions are oligosaccharides with varying chain length with an alpha configuration, which is a mixture of alpha limit dextrins, branched chain oligosaccharides of 6 to 8 glucose units, which may contain both 1,4 and alpha-1,6 linkages. Other amylolactic enzymes participate in the process of complete starch breakdown later on, but the contribution of alpha-amylase is a prerequisite for the completion of this process. Human alpha-amylase is a classical calcium containing enzyme. It is composed of 512 amino acids in a single polypeptide chain with a molecular weight of about 57.6 kd. There are 5 amylase genes clustered in the chromosome 1. 3 of them code for salivary amylase, amylase 1A, amylase 1B and amylase 1C, and the other 2 genes, amylase 2A and amylase 2B, are expressed in pancreas. Human salivary and pancreatic alpha amylase share high degree of amino acid sequence similarity. The second category of amylases is exoamylases. Exoamylases can break an alpha glucoside bond from the free end of an oligosaccharide. These are 3 types of exoamylases which differ in their mode of action. The first one is glucoamylase, it is also called as 1,4 alpha glucosidase. This enzyme breaks 1,4-alpha glucoside bond from the terminal end of an oligosaccharide with release of 1 glucose unit at a time. It cannot hydrolyze 1,6-alpha glucoside bond which is present at the branching point, and therefore, it leaves an isomaltose residue after the hydrolysis. Second type is beta amylase. Beta amylases also act on the terminal end of the oligosaccharide chain and break 1,4-alpha glucoside bond, but at an alternate glucose unit, thus releasing maltose and isomaltose as the residues. Beta amylases are present in yeast, molds, bacteria and plants, particularly in the germinating seeds. They are the principal component of a mixture which is called as diastase, that is used in the removal of starchy sizing agents from the textile and also in the conversion of cereal grains to the fermentable sugars. Third type of enzyme is isomaltase, or it is called as 1,6-alpha glucosidase. This enzyme acts on 1,6-alpha glucoside linkage which is present at the branching point in amylopectin or also in glycogen, and it is essential for complete saccharification of the starch. This is released by the secretory cells of intestinal mucosa, and it is also found in sweet potato, and it is also secreted by some of the microorganisms. Now, after learning about the amylases, let us see the process of digestion of starch in human digestive system. The first phase is salivary digestion. An alpha amylase which is called as ptyalin is produced by the salivary glands. Ptyalin is mixed with food in the mouth where it acts upon the gelatinized starch. Although the food remains in the mouth for only a short time during chewing, the action of ptyalin continues for several hours in the stomach until the food is mixed with the stomach secretions and the high acidity inactivates the ptyalin. Digestive action of ptyalin depends upon how thoroughly the food has been mixed with saliva, how much acid is in the stomach, and how rapidly the stomach contents are emptied. Under optimum conditions, as much as 30 to 40% of the ingested starches can be broken down to maltose by ptyalin during digestion in the stomach. The second phase is intestinal digestion. When the food passes to the small intestine, the breakdown of remainder of the starch molecules is catalyzed by pancreatic alpha amylase, which can also digest uncooked raw starch. This step occurs in the first section of the small intestine called as duodenum, the region into which the pancreatic juices empty themselves. The byproduct of pancreatic amylase hydrolysis, that is the dextrins and the oligosaccharides, are ultimately broken down to glucose by other enzymes which are secreted by the mucosal cells in brush border of small intestine, and later on they are rapidly absorbed through the small intestinal wall. The undigested starch, which is also called as the resistant starch, that passes into the colon and is later on digested by the microflora. So, dear students, in this part we have learnt about the basic structure of starch, mode of action of different types of amylases, sources of amylases, and the process of digestion of starch in human digestive system. Thank you.